Hello everyone, and welcome to the Society for Marine Mammalogy Seminar Select Series. My name is Luis Hochstad, and I am an Associate Editor for the Marine Mammal Science Journal. It's my pleasure to welcome you to this special talk today. The Society for Marine Mammal Seminar Editor Select Series highlights the latest and most exciting marine mammal science published in the Marine Mammal Science Journal. This is your chance to engage with marine mammal scientists, learn, and ask questions from anywhere in the world. As everybody knows, today is the International Day of Women and Girls in Science, and I couldn't be more excited to introduce our speaker tonight, Dr. Erin D'Agnese. Dr. D'Agnese is a postdoctoral scholar at the University of Washington with a specialty in One Health research, specifically using molecular methods for wildlife health and conservation. In today's seminar, Dr. D'Agnese is going to talk to us about the research she did as part of her dissertation at UC Davis School of Veterinary Medicine. She recently moved back to the United States from Tasmania, where she did the remainder of her PhD research in collaboration with the University of Tasmania on microbial connectivity between aquaculture salmon and wild fur seals. Her current work as a postdoc at UW is on eDNA in freshwater system, monitoring community changes during salmon restoration projects. In today's talk, Re-emergence of Guadalupe fur seals in Oregon and Washington, Dr. D'Agnese discusses the health risk facing Guadalupe fur seals as they re-emerge in their historic pre-sealing migration range. Before we start, just a couple of reminders for everyone. If you want to submit questions, please use the Q&A section of the Zoom interface. If you're on Facebook Live, you can also send comments and questions. We're going to also direct those to the speaker tonight. The article is available right now as an open access. We're going to be sending through the chat the link if you're interested in downloading the article. And with that, join me in welcoming Dr. D'Agnese.
Thank you. Thank you. Let me get presentation up for everyone. All right. So thank you again for inviting me to do this talk. This was very fun research to be involved with all of these lovely co-authors. And like you said, it was part of my PhD research. So always a good thing to revisit. So we'll be talking today about Guadalupe fur seals and their reemergence in Oregon and Washington. So just to acclimate everybody to fur seals globally, fur seals are a otariid within pinnipeds. So like sea lions and fur seals are all otariids. And they're interestingly, their undercoat, they have an undercoat with guard hairs, and that makes them very unique in that they use that for thermoregulation. Most species of fur seals occur in the southern hemisphere, like these guys. These are Australian fur seals around Tasmania. However, there are 2 species in the northern hemisphere. They do typically occur in colder habitats, so they're adapted for that with that thick fur. They do often breed on offshore rocky islands. However, they were hunted to near extinction globally, all species, for their fur. However, most species have rebounded pretty successfully. So let's talk specifically about Guadalupe fur seals. So they are fantastic species. They were presumed extinct essentially twice. So in sealing times, they were presumed extinct about in the 1800s. So early 1900s. And then 2 males were resighted in 1928, and then they weren't seen again and presumed extinct again until 1949. And then in the early 50s, a small breeding colony was found on one of the islands. Since then, their population has grown steadily from about 10 to 14% annually, which is great. They do remain an endangered species listed threatened species, as well as a NOAA listed threatened species, and they are still considered endangered under Mexican law. So the interesting thing about them is that there is a lot of archaeological evidence of Guadalupe fur seal bones on the most northern portion of the coast of Washington state. However, the first modern report of Guadalupe fur seals in the Pacific Northwest was in 1992, and then they started stranding regularly starting in 2005. So just to acclimate you with some, like the life of a Guadalupe fur seal, they breed on rookeries, primarily on 2 islands off the coast of Mexico. And pups tend to stay with their mothers for about 9 months until weaning. So like most fur seals, the pups hang out on the rookery while the female goes and forages and comes back and nurses and does that for a while until the pups are big enough, and then they start following them around. So then after they're weaned, they do tend to disperse away from their rookeries, and they do this in order to hunt for their preferred prey species, which are in the open ocean. So they're considered a pelagic species. They hunt things like squid and other open ocean species. So I want to just show you what we're going to be talking about kind of in the progression. So first, this was, First, we're going to talk about stranding response in general, and then that leads to some detailed necropsies and histopathology and ancillary diagnostics. And that's kind of the pipeline for how we determine cause of death. And then we also wanted to know whether there were any ocean conditions or external factors related to their stranding numbers. And so we did a regression analysis, which we'll talk about in depth. And specifically to the causes of death, we looked whether there were any spatial and temporal clusters, as well as considering ocean conditions as related to emaciation as potentially related. So when let's just start with stranding response. So there might be quite a few people that are familiar with what a stranding response entails. Often stranding response coordinators are contacted by the public or by the NOAA hotline, which are field reports from the public. And the responder shows up to a seal on the beach, and they take detailed notes on time, place, and circumstance and the animal demographic, and make sure that they're keeping track of exactly what information they can on that animal at the time. But they have also got an animal to deal with. So the animal response differs depending whether it's dead or alive. So if it's dead, often we collect the carcass for necropsy, especially if it's a Guadalupe fur seal in Oregon and Washington. And if it's alive, often they will provide assistance to the animal if needed. Sometimes it might not need it. It might just be, okay, let it rest, cordon it off, let it do its thing and go back into the water. Or it could be that it's entangled and needs something removed from around its body, or it may be fairly sick and needing some rehab assistance. So it just depends on the circumstances. We're going to talk mostly about the dead ones today, and all of the information that we can glean from them. But let's first discuss just pure stranding count numbers. So this is a figure from the manuscript, which encompasses the years from 2005 to 2016 of the number of Guadalupe fur seals stranding per year in the region. So interestingly, there was an unusual mortality event that was declared by NOAA in 2007 to 2009. And then you kind of see this trend as it's continued to increase over time. So an unusual mortality event is declared by NOAA, and it helps add resources and attention to a species that could be stranding in higher numbers at the time, or multiple species if it's a big event. So this species was declared, had a UME declared in 2007, 2009, like I mentioned, in Oregon and Washington. And then there was another one that was declared in 2015 in California, which is ongoing. So it's still an unusual mortality event time in California. And Oregon and Washington were added in 2019. So currently on the whole West Coast, California included, as per numbers I'm seeing, there's about 629 animals that have stranded in this UME between California, Oregon, and Washington. And then in 2019 and 2020, we had quite a few Guadalupe fur seals stranded in Oregon and Washington, 91 in 2019 and 42 in 2020. So I've extended that figure to include both of the UME timeframes. So you can kind of see that there is a general increase, but Guadalupe fur seals are definitely becoming more common to see stranding in Oregon and Washington. So the other thing we want to think about is, when are they stranding? So it's all good and nice to think about yearly trends, but we want to think about when in the year are they stranding. And if you've responded in Oregon and Washington, you'll know that between May and July is when we get the vast majority of Guadalupe fur seal strandings. And they typically tend to be yearling animals or weaned pups. So young animals, juvenile animals, that are showing up on the beaches, potentially not thriving in the region. So upon stranding response, often the bulk of our Guadalupe fur seals are found on beach surveys. So in Oregon and Washington, a lot of the coastline is drivable. Not all of it, but often we're going out and doing these driving surveys on the beach looking for dead marine mammals. And sometimes you might have a day like this where you bring back 7 Guadalupe fur seals. And this was a day in June in 2019. So when we're thinking about what factors might be driving these higher numbers during a certain time of year, during certain years, just general data analysis kind of happens in the same way no matter what. Like, you explore your data, you examine some relationships between your variables to see if you've got any collinearity or any issues that could be confounding. And then you determine if there's factors related to numbers using, in this case, we're going to use regression analysis. And the factors and variables we included were year, month, age class of the animal, sex of the animal, and some ocean conditions. And I'll go over what ocean conditions in detail. So quite a few, there's lots of words, we'll go through it. So the first one I'm going to talk about is Pacific Decadal Oscillation, or PDO, which is a monthly value calculated from the spatial average of sea surface temp across the whole Pacific Ocean. And then there's the upwelling index, which indicates the amount of upwelling that's occurring from deep, nutrient-rich water, bringing nutrients up to the surface. Sea surface temperature is a monthly average sea surface temp at a buoy, and we measured a buoy. We got measurements for all of these things from a buoy off of the Oregon coast in Newport, so it's kind of in the middle of the range, or it's as close to the middle of the region as possible. And then multivariate El Nino, or Southern Oscillation Index, we're going to call it MEI from here on out, is a bi-monthly time series index, which tells you whether you're in an El Nino, a La Nina, and it's based off a lot of different conditions. So sea surface temp, pressure, surface wind, radiation, all sorts of stuff across the whole tropical Pacific basin. And then we had some ecological integrity, matrices based on copepod biomass and community richness. So this kind of tells you what the productivity in the region. So if you've got lots of copepods, you often have lots of fish, might have lots of marine mammals. So we have northern copepod biomass, which are copepods coming down from the polar regions, and southern copepod biomass, which is them coming up from the tropics, and then community richness. And all of these are measures below or above the average, which is set at 0. So what we had is we had a lot of variables, and we had a relatively small sample size. So we had to reduce our variables for analysis. So one way we did that is testing for collinearity with a Kendall rank correlation. So if a variable correlated to 5 or more of the covariables, just the majority of them, they are removed from model testing. So after this, we were left with year, month, upwelling, southern copepod biomass, anomaly index, and copepod richness that we then tested for potential significance with stranding numbers with a bivariate regression analysis. And after that, we were left with year, month, and upwelling to be included in our full model regression analysis. And then because we had over dispersion of and an inflation of zeros in our data set, it was found that the zero inflated negative binomial regression analysis was most appropriate for this data. So there's a count model portion of the model and a zero inflated logit model portion of the model, which kind of discerns patterns where zeros are expected outcomes. So we used winter as the reference for that logit portion of the model, as it's consistently the season with the least amount of strandings and the most likely to have over inflation of zeros. And what we found is that we use seasonality as a known important factor for strandings for this species. And what we found was seasonality and upwelling were the two most explanative variables for stranding number increases in the region. So we also did, sorry, my dog is, there's a squirrel that runs around. Anyway, so we also wanted to look at whether the MEI variable was related, because it has been related to or implicated in Guadalupe fur seal strandings previously in California and Mexico. So, but it did have really high collinearity, but we tested it separately just to check. And we did find that through the bivariate regression analysis that there was no significant relationship between stranding numbers in Oregon and Washington and MEI. So there wasn't necessarily a relationship between El Nino and Guadalupe fur seal strandings in the region. Seasonality was a driving factor for stranding numbers. And the interesting thing about upwelling is that upwelling in the summer in the region aligns with currents from the north, also bringing nutrient-rich waters to the region. So you have nutrient-rich waters coming from underneath the ocean and them coming from the north. So it typically brings a lot of fish to the region, which are known to be exploited by a lot of marine mammal species. So it's not necessarily surprising that these animals are in the region potentially for the same reasons. So then we want to think about dead things. So we're going to talk a little bit about necropsies. Can't have a stranding response presentation without some necropsy photos. And so these are kind of interesting. The one in the bottom right is the first ever fur seal, Guadalupe fur seal, that stranded in Washington in 2005. And then this one on the left is one that Diana Lamborn and I necropsied this last weekend. So, you know, from the beginning to now. And this is an adult female that I responded to, and she was alive in around Christmas 2014. Yeah, right? Yeah. And so we're going to go into kind of the importance of necropsies, what we can learn, and how we determine a cause of death. So necropsies are the first step, gross exam of external and internal tissues. So we know lesions and abnormalities of tissues. And then often trauma might be very obvious upon necropsy, whether there's actually gear, like entanglement, net entanglement around the neck, or whether you've got gross amounts of hemorrhage in the tissues. We also take samples of tissues for pathology and molecular testing. And then that brings us to histopathology. So that's the microscopic exam of the tissues by the pathologist. And they note lesions and abnormalities within tissues microscopically. Sometimes you can see some pathogens directly in tissues, or there could be evidence of them, like inflammation, that you can see grossly, but you can see more directly in tissues. And often you can determine a cause of death from this. But if not, say you see some damage in a tissue, but you don't necessarily know what organism did that, whether it's a pathogen or it could be depending on toxin. Pathologists have, they're good at giving you an idea of what it is, and they're ordering an ancillary test for confirmation. So these are often microbiological or molecular tests for pathogens or toxins, contaminants, all sorts of things. So culturing bacteria from tissues, running PCR, qPCR sequencing to determine if DNA of a pathogen is present in the tissue, immunohistochemistry. There's all sorts of very useful tests that can give you a yes/no answer as to whether there was a disease-causing agent present, including tests for toxins or contaminants within tissues. So through all this, we ended up with essentially 3 leading causes of death categories. Emaciation as the most prevalent, with it being the primary cause of death in 41 animals. So when we do a necropsy, and determine a cause of death. Often there's a primary, a secondary, and a tertiary cause of death, if we can get to that level. And so there's 41 primary, and then we found that this in 69 of the animals, emaciation was contributing to the cause of death. So that includes the 41 primary cases, in addition to the other ones that were secondary or tertiary, deemed secondary or tertiary. So emaciation is a lack of fat in the tissues, lack of blubber. And then we also had trauma and infectious disease. And I'm going to talk about those a little bit more in depth. But we had both human-related trauma, potentially, which was the primary cause of death in 17 animals, unknown sources of trauma, and then predation. And infectious disease, we had 18 that was deemed the primary cause of death, but interestingly enough, it was the second most contributing cause of death. So 49 animals had it as a contributing cause of death. And the most common one we found upon testing was protozoal parasitic diseases, which we're going to talk about in a second. We also found GI parasites, had some bacterial sepsis, hepatitis contributing in one case, and some quite a few unknown disease processes. And it can be unknown for a whole myriad of reasons. Degraded tissues, not able to really isolate something from it because of degradation and all sorts of things. So for the cause of trauma, I just want to break it down. So we had entanglement and fisheries related trauma in 13 cases, contributing in 14. And in some cases it was confirmed, which usually entails finding gear on the animal. Also, we had many that were suspected from lesions upon necropsy, which are usually you'll have lateral like hemorrhage that looks like it's had something pressing into its tissues and causing bruising. Or it could be a laceration in the dermal layer, so in your skin, all sorts of things. And then we had blunt force trauma in 11 cases. It was suspected human related in 3, but often you can't confirm that without any gear, or without any direct knowledge of what happened. But it was a primary cause of death in 8 animals from an unknown source. Then there was 2 that were shot, and 1 that was predated by a shark, and an additional 2 that had bacterial sepsis as the primary cause of death, but that was likely the bacteria got in likely from these penetrating lacerations that were found. So we're also going to talk about the parasites we found. So protozoal parasites were suspected as a potential cause of death upon necropsy in 30 animals, and was found positive in 22 of those animals. It was the primary cause of death in 4 seals, but contributed to death in all 22 of those cases. So, or contributing to death in 18 cases, sorry. So these include Toxoplasma gondii. In 15 seals tested positive for that, and 11 seals tested positive for Sarcosis cicerona. Interestingly, there were 10 seals that were co-infected with more than one coccidian parasite. Usually it was T. gondii and Sarcosis cicerona, but 3 cases tested positive for Neospora caninum, along with either S. neurona or T. gondii. And then we also had quite a few gastrointestinal parasites, which were observed in 21 Guadalupe fur seals upon necropsy. Generally, the level of parasitic infection was fairly low, but this is likely underrepresented because they are quite hard to see upon necropsy. And Guadalupe fur seals have fairly delicate GI tracts, so it's hard to kind of sift through them as easily as it would be in a larger species. So we wanted to see whether there were clusters of causes of death, and they were, we used 2 different models. So we did a Bernoulli model of spatial temporal clustering for each cause of death individually. And then we also did a multinomial model that we used to find co-occurrence of cause of death in space and time. So we had 2 significant clusters, one with the Bernoulli method of trauma cases around the mouth of the Columbia River, where there's high levels of commercial fishing, and one of both emaciation and infectious disease cases a little further north around Grays Harbor Ocean Shores, Washington. So we wanted to know whether potentially ocean conditions could be related to a cause of death, and the only one that had any sort of relationship that made sense was emaciation. And we performed logistic regression analysis for emaciation, ocean conditions, and the most explanative model for that was emaciation related to Pacific decadal oscillation and copepod richness. So a high PDO means low productivity in the region. So a higher PDO leading to an increased odds of death means that there was lower productivity in the region, likely leading to reduced prey availability potentially. And then copepod richness is like the opposite relationship. But you'll note neither of these values were actually significant. This was just the most explanative model. So likely this is an artifact of sample size, not having quite enough. It could be any number of reasons, but it is interesting, is something interesting to think about as we move forward. So the big main findings, the vast majority of Guadalupe fur seals in Oregon and Washington are juveniles. We believe they're likely migrating post-weaning for resources, and their strandings are very seasonally linked and related to increased upwelling in the area, so increased amount of nutrient-rich water coming up. The clusters of trauma near the mouth of the Columbia River were high, which is interesting, especially in consideration with the commercial fishing that takes place in that region, especially considering that many of the trauma cases were related to entanglements and fisheries interactions. So it appears that competition and interactions with fisheries are placing Guadalupe fur seals at a higher risk in this region, which is pretty common for the species throughout their range. The cluster of emaciation and infectious disease cases near ocean shores and Grays Harbor is fairly interesting and could indicate that as they get further up, they're using resources, it's getting colder. It could just indicate that there's a reduced fitness when they reach further north. The other thing is that with emaciation and infectious disease, you don't necessarily know which one came first. So if they've developed an infectious disease, it typically makes them less robust and able to forage. They get emaciated. And if they are having a hard time foraging, they're emaciated, their immune system suppressed, they often can succumb to infectious disease more easily as well. So that relationship is hard to tease out, and just something to keep in mind, especially when there were so many that were both emaciation and infectious disease. So for the future, it's likely that they're going to continue to strand in Oregon and Washington, increasing numbers. That's what we've been seeing. And this is supported by having sighting data. It's not included in the manuscript, but there is sighting data from marine mammal observers, which indicate that they do successfully forage offshore. So this pup on the kelp is one from offshore in 2011 that someone witnessed. And there's been multiple sightings. Sorry, my dog just distracted me. There's been multiple sightings of these guys off the coast. We know that there's a risk of interactions with fisheries and becoming entangled, and that likely remains high. So it's something that we should continue to monitor and manage and mitigate through management decisions. But their presence in Oregon and Washington, which is interesting, doesn't appear to be related to extreme weather events or any one factor. So we believe they're here, and they're likely here not to stay because they come up and leave, but they're likely here and are going to continue to show up. So like I said before, these are offshore animals. They should be offshore foraging. They should not be on a beach. And if you happen to be on the coast and see a little brown furry cute thing like this, it's always a good idea, no matter what, to call the NOAA Stranding hotline, which I put the number down here. We can replay it as many times as you want, write it down, keep it in your wallet, wherever, or on your phone, I don't care. But it's always a good thing to have at hand in case you do see that, because they will tell you whether they know about that animal. They can direct stranding response coordinators to it. These animals are quite delicate and can be quite feisty. So if you do see an animal with net around it, it's always better to have someone that knows how to handle marine mammals go respond than to be the, to do the Good Samaritan thing, but also Good Samaritans are great. So don't want to discourage that, but definitely handling marine mammals comes with like experience. So the other thing that I just want to end kind of on is that this is actually kind of a happy story of conservation success. So whilst we talked about a lot of dead things and a lot of health risks for these animals, the kind of take-home message is that necropsies are really useful tools for measuring populations and their presence in a region, especially if that population are animals like Guadalupe fur seals that are mostly offshore and you don't see regularly, but they're there. And it will help us, and it always helps understand what health risks they're facing through necropsies. So Guadalupe fur seals are likely stranding because their population is getting healthier and healthier and increasing. And this ongoing UME research that both California and Oregon and Washington are doing, and NOAA's helping lead, will help elucidate if there's any underlying factors which are leading to increased numbers as we move forward. But it really appears at this stage that conservation efforts in both Mexico and the US are working for this population, and that's really great news. So yay for Guadalupe fur seals. And lastly, I just want to thank everybody that I've worked with along the way, my PhD supervisor, Valtrina Smith at UC Davis, my colleagues, collaborators at Washington Department of Fish and Wildlife, especially Diana Lamborn, and our collaborators in the Pacific Northwest, Portland State University, Oregon State University, Cascadia Research Collective, the Makah Tribe, Tribal Nation, for all the stranding response they do. I want to make a special shout out to all Marine Mammal Stranding Network volunteers globally. I'll just do globally, especially the ones in Oregon, Washington for this research, but we wouldn't be able to do what we do, and we wouldn't be able to do the science that we're able to do and get the information we are able to get without volunteers. If you want to volunteer, I'm sure there is a stranding response network somewhere that would be happy to have you. And I'd like to thank our funders and my friend Jeff Harris with NIML for all of the beautiful Guadalupe fur seals that peppered my presentation, other than the dead ones, which are mostly from Diana, which also thank you, Diana. So yeah, now I'll just open it up for the Q&A portion.
Thank you so much, Erin. That was a really, really good talk. Really interesting data you have. For everybody that is watching us on Zoom, we have a little glitch with the Q&A, but it's not working. So please, you can just post your questions in the Q&A rather than in the chat. And for those of you that are joining us through Facebook, please ask your question in the comments. We also have to thank the student members that are reading through your questions, and they're going to help us a little bit with the conversation right now. I do have a couple of questions, but I'm just going to start with one at the beginning, and then you'll see, we'll see what everybody else is asking. But the question refers to the life cycle of the fur seals, the Galapagos fur seals. The fact that you're seeing these animals arriving in May, July is kind of the summer, but can you comment a little bit in terms of when do the colonies break up? When are you expecting these animals to leave from Mexico and start spreading in the ocean?
So they should be weaning around like the Marchish, February, March times, if I recall correctly. So it's been a while since I thought about this, but if I recall correctly, they're weaning in about that springtime, and then they essentially be going up directly. So it would only be a few months before they start showing up where we are. So they breed in the summer months, and then there's about 9 months, and then they're up there the next summer. So that's why in May, they're not quite the young of the year. They breed in like June, July. So they're considered wean pups still in May, but once that transitions over to June, July, they're essentially a yearling. That's how we denoted that difference.
Okay, I'm gonna open up the floor for questions because there's a lot of questions that are arriving right now. So I'm gonna try to read a couple of them, and then hopefully the students are gonna help us with the questions from Facebook at the beginning. Sure. The first question from Ignacia Mesa is, she's asking whether hookworm parasites have been identified in Guadalupe fur seals. Yes,
the answer is yes. Yeah, we found hookworms. We've even had an Anisakis complex things. There's quite a wide variety of of parasites that we found, but hookworms were one of them. Yeah.
There's another question about what is the contaminants that you usually test these animals for when you're doing the necropsies?
There wasn't a lot of blubber on these animals, so contaminants wasn't done as regularly. We did test these animals for domoic acid, quite a few of them, and we just didn't get any that were really that significantly positive. I think there was one animal that had one or two animals that had a couple like higher levels of domoic acid, but I don't think that we really tested contaminants on these animals, mostly because of the tissue degradation and how expensive it is to run contaminants. And we just didn't really, they were really emaciated. We didn't have a lot of blubber. So that would be an interesting thing to do, but likely hard to do on Guadalupe fur seals in Oregon and Washington, where they're highly emaciated.
I have a question about the fact that you found toxoplasma in these animals. I'm not a parasitologist, so I'm not really familiar with that, but are toxoplasma related to humans? Basically, is that the source of toxoplasma in the marine environment?
So toxoplasma in the marine environment, there's a lot of research out there about toxo in the marine environment. And it comes from cats, like toxo reproduces in cats, right? And then they shed it out, the oocysts, the eggs of the toxo, they shed it out in their feces. And then they're super persistent in the environment. And it gets runoff from land into rivers and out to the ocean and bioaccumulates, at least in like filter feeding bivalves, because that's how the sea otters, not just bivalves, filter feeding mollusks, so snails and also bivalves. And that's how sea otters were getting it. There's not really good information on how it's necessarily getting into things like fur seals that aren't eating. They are not eating filter feeding like bivalves from the ocean, like from the floor near shore. They're eating squid and fish offshore. So it's an excellent question. We would love to know the answer of how it is that toxo and sarco, that are land-based pathogens, are getting into fur seals and sea lions. It could be that there's a variant that's actually shed by sea lions. That's a potential that I think there's a few people working on that. There's definitely people out there that know much more about what's going on in the toxo world than I do, but it's an excellent question. I hope we find out.
I think we should probably open it up for Facebook. I don't know, Teresa, if you have any questions from Facebook for Erin.
This is Charles in lieu of Teresa. There's been some technical glitches. There are nothing on Facebook just yet. There was one, but it was a question just generally about if this was going to be shared broadly. So it's just a reminder that this will be recorded and available on our YouTube site next week. And you can always see it on Facebook replayed. So you can keep going with the list of questions, Louise, if you want.
Okay, yeah. So there's several questions about the range of Guadalupe fur seals in the ocean. How far north do they spread? Do you know the distribution at sea of these animals?
See, I think there's even been reports. Oh, I don't want to say this and be wrong. It's been a minute, but they go really far out. There's a lot of good telemetry data about how far some females go offshore, and I just don't have that number off the top of my head. Sorry, but I think I remember there being. No, I'm not. I don't even want to go there because I just vaguely remember someone saying that like Hawaii one showed up at some point, but that's probably not a good thing nor a normal thing. So
wasn't that the domoic acid case?
Oof, not one of ours. So I don't know, maybe. Probably, I don't know, who knows. But yeah, they are really like far moving animals. And I just wish I had that like kilometers in my brain, but I don't. I'm sorry.
Scott Peterson is interested in knowing how do you tease apart disease from emaciation in your animals? How can you tell what's the cause of death ultimately?
Oh, how do we, can you repeat this?
How do you disentangle disease and emaciation in your animals? Is it lack of
how
fat
do we
what causes it?
disentangle disease and emaciation? Okay, so that is having good pathologists is helpful. So what we kind of did is emaciation was almost like universal in these animals that we're finding. They all had reduced blubber thickness. They all, not all, like I don't want to say all, but the majority of them had reduced, it's like fat in their tissues. So pathologists can look at the tissues and see if there's lipids and other fats, well, lipids in there, like around their cells. And there should be quite a happy amount of that in the tissue, as well as some like mesenteric fat and some other fat. So you start with that. And then essentially, if there's no, upon necropsy, there's no evidence of infectious disease, then the leading cause of death is emaciation. Then there's different degrees of infection, like inflammation and infectious disease processes within an animal. So if there, you do find, say, like a toxo case. So we know that there's toxo there, there's little lesions. We found it through PCR, but there wasn't really any big lesions that would cause like death. But they were severely emaciated. They were definitely not having the nutrition that they needed to survive. That was often declared the leading cause of death, and infectious disease was secondary. If the infectious disease process was something that could acutely cause death, it would have been termed the primary cause of death. Does that answer the question?
Hope so.
Like,
so going back to the topic of Toxoplasma and the anthropic origin of these samples, we're going through our regular pandemic of the year, right?
yeah.
But thinking a little bit about the impact of humans, diseases and parasites and whatever in wildlife, marine mammals. For a previous pandemic, for the swine flu, for H1N1, they actually found the virus in elephant seals, in northern elephant seals of the coast of California. So how do you think something like this could potentially be affecting Guadalupe fur seals?
Like COVID?
Yeah.
Oh. I mean, there's definitely viral particles that are being shed in sewage and getting into the, like in stormwater and getting out to the ocean. I assume there are, I think there are people that are looking into, you know, COVID in wildlife. I remember talking to someone in Australia that was doing some, you know, monitoring that. I could see a case where, you know, there are potentially, it's a zoonotic pathogen, you know, it's a zoonotic pathogen that, you know, animals in the wild environment could be interacting with these viral particles. Another, you know, if it's able to kind of replicate in a tissue, in a fur seal, maybe not cause disease, but could replicate and mutate and eventually end up, you know, having a fur seal COVID. I mean, there's no reason that it couldn't, but I don't know that there's any evidence yet that it's impacting wild populations.
And it's perhaps also sort of a warning story too for people not to get closer to these animals if they found one in the wild. You don't know what kind of diseases you could potentially transmit to a population that is in danger, just recovering. Right?
Yeah. I mean, these guys, because they're still recovering, are at risk for a lot of things. You know, if they did encounter a disease. So one of the reasons we did this initially wasn't, you know, to prove that they're here. We're like, okay, they're here. Why are they dying? Is there something big that we need to be worried about? Are they getting a novel disease? Are they getting a disease that is potentially novel to them and would be super risky for them because they are a naive population to that or not? And we just found that the not is the answer for this particular population right now with the diseases that are out there. But that doesn't mean that if something like COVID or something that is novel enters the environment and you have these endangered species that, you know, breed in rookeries where most of the population's at 2 islands. So if something were to happen in the population, they go back and they spread it around essentially all of them in the population, that could cause like huge issues for the population, for the species as a whole, even because they really are primarily breeding on 2 islands. So if something even happened to one of the islands, like an earthquake or anything, like this population's a pretty, you know, at a very testy place. So until they start really breeding elsewhere, and like spreading out a little bit more in those close quarters, they are at risk of spreading disease.
I'm going to stop asking questions because there's a lot of other questions, and I want to give people the chance to ask their questions. So Daniel Iris is asking whether you have any thoughts about which commercial fisheries around the mouth of the Columbia River are impacting these animals. Your dog is one. Just a minute. She has a dog and the dog is losing it a little bit. She'll be back. In the meantime, if you guys are interested, the paper is open access right now. You can find the link on the chat box, so you can download it for free right now. And we're receiving comments too through Facebook. So if you're interested in sending questions through Facebook, not on Zoom, please do.
Sorry about that.
Yeah, no worries. So the question was about commercial fisheries around the mouth of the Columbia River. Which ones do you think are impacting these fur seals?
Essentially anything that's doing net fishing out in pelagic areas, and so could be caught, like putting them at risk. We do find quite a lot of, I'm not the best at which net's which, but I'm pretty sure it's gill net, that we find on these animals, which is a very predominantly used method. And I mean, they're targeting mostly squid and offshore prey species. So if they're targeting those species and commercial fisheries are targeting those species as well, then there's likely overlap for interaction. And I would say those are the most likely ones. But I don't want to, like, you know, say all fisheries are bad. Oh, trawl netting. Diana just texted me. Trawl netting. Thank you, Diana.
Conrad Erklens is asking whether you normalize your level of effort for the beach service looking for strandings.
Say that again, sorry.
Whether you normalize the level of effort for the beach service.
Oh, so there, Jesse Huggins, who's on this paper, actually published a good paper about how much better beach surveys are if you do them regularly than even like reportings from the public at capturing, especially Guadalupe fur seals. These things are very underreported in the region. So I feel like typically there's beach surveys being done, you know, at least a couple of times a month during the summer, if not more regularly. But it depends on the stranding response, coordination. It just depends on what's going on. There could be all sorts of, you know, they're busy. Summer is a high time for all sorts of strandings. I know in Washington, it's the peak season for harbor seal pups as well in the inland waters. So like, it's, you know, stranding response networks are usually stretched fairly thin, but there is really good, like normally done beach surveys that are going to capture most of these animals. There is like a whole portion of like Southern Oregon coast and the northern portion of the Washington coast, the Olympic Peninsula, is not drivable. So there's likely is like some bias in our clusters that they do tend to be located around those areas where we have beach surveys, but we did have lots of animals all over the place. So it might have moved the cluster a little bit more further south than it would be usually, but it wouldn't have really changed the cluster that much, is what we kind of saw in our data.
Sarah Grimes is asking how many adult and subadult animals are stranded.
Let me pull up that. There was, how do I get out of here? There we go. Let me just double check before I give you a number that could be wrong. So we had a total of 9 adults that stranded. That was it. And they were spread out pretty evenly amongst all the seasons. So we had 2 in winter, 2 in spring, 3 in summer, and 2 in fall. And we didn't have any subadults.
So Denise Gregg is asking, following on my question about toxoplasma, do you see more toxo during marine heat wave years when fur seals are pushed closer to the shore?
I didn't actually look at that. I need to look at that data again. That's a good question. I'll check.
Someone's asking, Yuri Fernandez is asking about the presence of cats on the islands where these animals breed.
Oh, on the Rookery Islands? Are there cats on the Rookery Islands? I don't know the answer to that, but I would imagine not.
No, there are a lot of, well, there's actually a lot of.
I don't know.
Cats, yeah.
I was like,
cats
I don't know.
In Guadalupe, actually, they've been reported to even steal milk from the elephant seals.
Really?
Yeah, I've been on that island. We were doing a project with elephant seals, and I saw them. Like, when you step out of your tent in the middle of the night, you see thousands and thousands of little eyes looking at you.
Oh God,
creepy. There's
that's
lots of
not
cats
ideal.
in Guadalupe Island.
Well, I'm glad that you're on because I'm like, I don't know. I mean, yeah, I guess there are cats pretty much almost everywhere. So that's good to know.
It's a big issue on that island in particular. I don't know about the other one.
Are there a lot of mice?
Is asking whether do you see any Guadalupe fur seals with patchy loss of guard hairs?
Oh, like alopecia?
Yeah.
I don't recall any. We get a lot of gooseneck barnacles on these guys. So there are quite a few cases with gooseneck barnacles, and they were more prevalent early, like 2012. There were quite a few, I think. But I don't recall many cases of alopecia in these guys. Like I've seen it a lot in Australian fur seals, but I don't recall any cases of alopecia in this one. There might have been a couple, but not noteworthy. I need to check on that. Diana probably knows.
Monique Lance is asking, what about the prey? Do you find any prey on the GI tracts of these specimens?
Yeah, we did. So we found quite a few squid beaks. Diana says only a couple alopecia cases. She's luckily listening and texting me, so that's nice. We found quite a few squid beaks. There were a couple cases where we had like plastic, like there was styrofoam in a couple of animals. There were they mostly didn't have much in the way of prey parts in their stomach. There were some otoliths. I think they were identified, but I don't remember all of the species that were listed. I think there were some that had herring and some sardine, but mostly when we did find food contents, it was squid for the most part.
Denise Schultz is asking about information for the last one that was collected last week in ocean shores, age-wise. Do you know anything about the animal last week? Oh,
that was the one that I believe that's the one that we ended up doing the necropsy on in. And it was an entanglement case, and upon necropsy had froth in the lungs and everything, quite a bit of evidence of drowning. But it was alive, it was seen alive on the beach, and then some officers removed the net off the neck, and then it subsequently died. And then we picked it up, and Diana invited me to come to the necropsy. So I did.
Let me see. There's a lot of questions that are getting here right now. And
Yearling.
Pabs
I was
is
a yearling. That one was a yearling. That's right. Sorry, that was
okay. Yeah.
sorry.
And Pabs is asking, she's saying that this is a very interesting presentation. She's thanking you, and she's asking if you could comment on whether your data suggests that there are any interspecific interactions with other ororites that might contribute to the distribution and or mortality in Washington or in Oregon. Is there anything like interactions with northern fur seals, with California sea lions?
Not really. I mean, there is like on San Miguel Island, there is a few Guadalupe fur seals that hang out on the same rookery as California sea lions. In Washington and Oregon off the coast, they're out offshore. So we don't really know, but how much they overlap in habitat when they're out foraging. But there's every possibility that if they're all targeting the same species, there is some interactions happening there. But when they're off the Oregon and Washington coast, they're mostly off foraging. It appears. I mean, we assume they're trying to forage.
Do you find many northern fur seals stranded as well, or just mostly Guadalupe fur seals?
There were more northern fur seals before the Guadalupe fur. There was a little bit of overlap early in the data series, but I think Diana hardly gets, she's the Marine Mammal Stranding Coordinator for Washington, and that's for DFW, and that's why I keep referring to her. She doesn't get a lot of northerns anymore. I mean, occasionally, but there's not a huge amount of overlap.
Fred Norton is asking whether you know any of the causes of the UMEs. Do you have hypotheses for this?
Well, one is very long. So the 2015 one to now is very long, and it's the bulk of the animals are in California, and I haven't seen their data. So I hazard to, speculate. For the one in 2007 to 2009, most of them were emaciation. So they're included in this data set. And so the findings kind of suggest that they were up here for the first time. But everything is still kind of under investigation as to the cause. It's very hard to discern one cause, but so I hazard to speculate. Let's just put it that way.
That's fair enough. Daniela Bernard is asking about collaborations with Mexican scientists. Is your group collaborating with Mexican scientists on this research?
I've spoken with some of them at conferences, and they're lovely, and I would love to collaborate with them more. Let's just put it that way. So if any of them are out there, then feel free to email me.
So Hilda Chavez is asking whether are there any initiatives to avoid entanglement or the interaction with humans? Is there anything that you're aware of that is happening?
Any management things that are happening or any interventions?
Yeah.
None that I know of directly off the Oregon and Washington coast. There might be, but that's not my area of expertise.
Okay, Eric Archer is asking, his question is, a lot of the southern cases have undetermined as COD. Are these removed in the model or treated as another category equivalent to emaciated or other determined categories?
No, in the model, we only included ones we had known causes of death for. So, because I didn't want to, because if you don't know whether it's not, then you can't say if it's not for sure. So we didn't include the undetermined cases in there, hence why the model was a little harder. It was our sample size was low because of that. And those undetermineds are a mixture of down there, surveys aren't happening regularly, as regularly, sorry. And so often they're coming upon these animals after they have decomposed quite heavily, and there's not really a lot of you can't really determine a cause of death if you don't have a fairly fresh carcass. If it's like mummified, or if it's just, a lot of them were just pelts and bones down there as well. So that's kind of it would be great to have more of an effort that's widespread, so we could see what's going on down there a little bit better.
Scott Pearson is asking whether the increase in strandings could be explained by population growth rates alone.
I'd say that's the most likely explanation for their increased stranding numbers, but I don't think it can be I'm not sure if it could be the only thing. Yeah, there's just so many climate-related variables that are changing. There's prey availability in the California current and down near their rookeries, that if there's low prey availability, they're forced out. So it's hard to know whether it's like that is what's like the most explanative thing, in my opinion.
Erin D'Agnese is asking whether pups and juveniles typically migrate north to Washington or Oregon, or do you know anything about the dispersion patterns of juveniles and pups?
Well, from the data we're seeing in this, it appears that is maybe not like all of the population does that, because there are some that go south. There's quite a bit of evidence in Mexico that they're going all over the place down there as well. So I wouldn't say it's inherently like the cool thing to do for Guadalupe fur seal, but it seems to be a normal thing for them to do if they want to go that way. I don't think I think the evidence that we've presented here suggests that it's a fairly normal thing that they're showing up. Like it was normal pre-sealing, and it seems to be becoming normal again.
And I don't know how many of these animals are tagged at their rookeries, but do you know whether animals come from Guadalupe or from the other islands, or there's no information on that?
No, we didn't have, I don't think we had a single tagged animal show up in our data set. So we don't know.
You don't know where it comes from. And the other question that I had was, do you know how your data compared to strandings in California or in Mexico?
Up until 2015, when the UME occurred in California, they were actually getting less strandings in California typically than we were in Oregon and Washington, and then it swapped. So that's an interesting thing that we still don't know. So that's still under investigation of why that shifted and why their numbers greatly increased after 2015. So yeah, they had a handful of cases. I don't want to say that they didn't have cases in the 2005 to 2016 range, but 2015 range, but they saw a significant uptick in 2015, and hence the UME.
Judy Fernandez is asking whether you found domoic acid on your animals.
We found it in a couple of animals, but I think only one was it actually related to cause of death. Then again, we did not test all animals because often there were like hardly any feces, hardly any urine. There was not a sample to collect to test for domoic acid.
Trying to get organized here and see what other questions there are, and there's a lot of questions still. Daryl also is asking whether you can determine whether the malnutrition stems from poor nutrition pre-weaning, or is something that happened while these animals were at sea.
The toxa? Wait, say that again.
Yeah, whether malnutrition stems
malnutrition.
from poor nutrition pre-weaning, or you think that this is something animals that were weaned at regular mass and they just weren't successful at sea?
I would say the second, but that's not necessarily, I would guess, because it's a wild animal, you never know. But reaching weaning is hard. Like the whole time being a juvenile seal pup in the wild is difficult. Prey availability for your mom not getting back in, prey availability for your mom could lead to not getting enough nutrition. Your mom not coming back regularly could lead to you not having significant nutrition. So there's a lot of different variables that make it very hard for a pup to even reach weaning. And then to have to, but the idea is that the whole time nursing, they're getting fat, and that's the whole point, right? So you hope that by the end, they're set up for success, but you don't know. And I don't know. The researchers in Mexico that are doing more work with the pups on the rookeries would have a much better understanding of that. And I just don't know. So.
Kim Aikman asked a question about whether there's a good upwelling in the area where the animals were found. Do you think that the upwelling conditions could have an effect on these strandings?
I mean, the reason we're looking at upwelling as an ocean condition is it's used as a metric for nutrients being upcycled and leading to higher productivity in the region. And that's what most, like when thinking about ocean conditions as a ecological indicator, is what it's mostly used for. So I don't know whether there are other things that could relate between upwelling and stranding, but my guess is that it's more indicating, it's more of a kind of indirect relationship of there's upwelling occurring, and that's driving a lot of productivity in the region, leading to increased prey and increased presence of all marine mammals, including Guadalupe fur seals, is what I would guess. But I don't actually know that.
So this other question is sort of somehow related, because Rudyard Wallen is asking whether there's any relationship between your strandings for Guadalupe fur seals and the trends that people have observed on brown pelicans or Hermann gulls breeding failure.
Ooh, I don't know, but I want that's interesting. I haven't thought about that. It's a good question. If I saw some trends and kind of saw those and had a better understanding of what those trends were, I could maybe weigh in on that, but I don't have that information. Sorry. Good question. Might look into that.
Yeah. Paul Nader is asking what predators are preying on Guadalupe fur seals.
Mostly sharks. Yeah.
Let me see. Someone is commenting that they've recovered seagrass and pipefish from a couple of Galapagos fur seals, Guadalupe fur seals pups. Oh,
yeah. I think we found some random detritus and plant matter in their stomachs as well. But you get hungry, you get desperate.
Then the same person, Sue Pemberton, is saying that animals were tagged in Mexico back in 2014, and we saw about 7 of those in Central California.
Oh, that's cool.
Yeah.
Yeah, they don't seem to be getting that quite as far north for us.
Ignacio Mesa, this is one of the last questions that we have, is saying that you mentioned the colonies are in rookeries, so he was wondering if that might be limiting the hookworm prevalence in your samples.
Yeah, I mean, that's the typical route for hookworm is through feces on a haul out. So yeah, they're likely encountering them, would be my guess at the rookeries.
There's a new question. Let me see what it is. Saying the Guadalupe fur seals are also moving over 1,000 kilometers south of Guadalupe and San Benito's Islands,
yeah, and
forming new haul-out colonies in the Gulf of California. It was Jan Heka or something like that.
Thank you for the number. It was killing me.
Do you recommend any regulations to the fishing industry?
Do I recommend any regulations to the fishing industry. In an ideal world, I would probably, but food security for humans is another huge issue. So that's tricky. So how do we catch, I mean, that's the ongoing thing with interactions with marine mammals, right? With fisheries, is how do we limit interactions with marine mammals whilst also, you know, feeding a growing human population? I think there are potentially tools, deterrents that you can use that might deter some animals, which would be great to start implementing off of boats. So this can be noise, this can be, you know, low-level sonar, you know, other things. So, you know, I would recommend, you know, potentially exploring more deterrents or coming up with new innovative deterrents that that might work better. I don't know what a Guadalupe fur seal would respond to as well as like some other species. So that's tough. Like limiting the amount of trawl netting and using other forms of fishing, so like long line and other things that are a little less invasive to the whole population might be useful. Yeah, there's all sorts of, that's an ongoing thing that I still am like, I don't know how to solve.
So I know that you have in your map that cluster of animals that were emaciated, but do you see any latitudinal trend as animals move farther north? Do you see more levels of or poor body condition?
Trying to think. I think I mean, in general, that cluster moving indicates that, but I don't know about further north. And also, I mean, the ones in Oregon are also pretty emaciated. So degree of emaciation wasn't something we really measured or kept, you know, that kind of information in our data set. So I don't actually have that answer. Sorry.
That's fine. I think that's it for questions. I don't see any newer questions popping up. I don't know if there's anything on Facebook. No. Sorry.
Oh, I left that on. I should have gone up one. There we go. Sorry.
I don't know if someone.
No new questions on Facebook right now. It seems like we're good there.
Yeah, I think we are good to wrap it up, Louise.
Thank you,
Perfect.
everybody, for all the good questions.
And thank you for giving such an amazing talk. It was very, very interesting, the topic that you were looking at, and how we can learn from Stranding is something that people should be more aware of. It's not all about live animals all the time.
Yeah.
So we would like to thank Erin for her talk, and stay tuned for the next seminar that is going to happen. It's going to be announced through the Society website and Facebook too, and Twitter account. So goodbye, everyone.